Hello. Today I will be talking about the short and long reflexes of the enteric nervous system. My name is Lily Solomon, and today is July 31st, 2024. I will first begin with a definition of the enteric nervous system, or the ENS. The ENS is a neural network within the gastrointestinal tract that functions independently and controls various aspects of the GI physiology. It is essential for regulating motility and secretion. There are many similarities between the ENS and the CNS, or the central nervous system. First is the intrinsic neurons. These neurons lie completely within the walls of the gut. Next are the neurotransmitters. The ENS neurons release more than 30 neurotransmitters that are identical to the ones in the brain. Some of these include serotonin, vasoactive intestinal peptides, and nitric oxide. Glial support cells are similar to astroglia of the brain, and diffusion barriers are not very permeable and are similar to the blood-brain barrier. Integrating center originates in the GI tract and integrates without leaving the ENS and is very similar to the brain and the spinal cord. So basically, the enteric nervous system is all in the GI tract and it tries to keep everything within the GI. So this leads to short reflexes, which are more locally controlled. This is like a little brain. So instead of having the brain and spinal cord, this is like a mini brain for the digestive system. The enteric nerve plexus and the gut walls act as a little brain that allow local reflexes to begin, to be integrated, and end completely in the GI tract, as I was saying. Short reflexes originate within the enteric nervous system and are integrated there without outside input. There's submucosous plexus. This contains sensory neurons that receive signals from the lumen of the gut. It controls secretion by GI epithelial cells. There's also myenteric plexus neurons that influence motility. Next is long reflexes in the integrating in the CNS. So rather than everything happening in the GI, it also sends information to the central nervous system as well. The ENS sends sensory information to the CNS and receives input from the CNS through autonomic neurons. Long reflexes are the GI reflexes that integrate the CNS rather in the ENS. Cephalic reflexes are things that happen in the brain. So if you think about something, you might be more hungry. So this is like feed forward reflexes. This is stimuli such as sight, smell, sound, or thought of food, and they prepare the digestive system for food that the brain is anticipating. So let's say that someone has cookies in the oven and it makes your house smell like cookies. If you really like cookies, this could make you salivate and this could make you think about eating food and lead you to be more hungry. Additionally, if you see a Thanksgiving meal, let's say, you might be anticipating to eat, and so you get more hungry and your body is ready for it. Finally, is emotional reflexes. Their influence on the GI tract illustrates the link between the brain and the digestive system. So basically, I know that there are some people who, if they're sick, they might not be as hungry, or if someone has anxiety, they might want to eat more. So emotionally, that could also link to hunger and the GI reflexes happening. Here is a larger demonstration of the integration of the digestive reflexes. Starting at the yellow, which is the stimulus, there are local stimuli such as the presence of food or cephalic, so things that are happening in the brain such as sight, or smell, or sound, like we said before. And this goes to the sensory receptors that lead to the little brain, or as we also know, the short reflexes that output a signal to the enteric neurons or the GI peptides. And this then goes to the target organs such as smooth muscle, the exocrine cells, the brain, etc., that go to the tissue response. These include changes in GI motility, the release of bile and pancreatic sensations, enzyme, acid, and bicarbonate synthesis and release, hunger and satisfactory rate, insulin and glucagon levels increasing and decreasing. Next, we look at GI peptides, including hormones, neuropeptides, and cytokinesis. GI peptides excite or inhibit motility and secretion. They also act outside of the GI tract, and some of their most interesting actions are in the brain, such as the CCK enhancing satisfactory rate. Ghrelin is also another, and it secretes in the stomach and acts on the brain to increase food intake. Researchers have had more than 30 peptides in the GI mucosa, and there's few peptides that are well defined in the pancrean effects. Non-peptide regulatory molecules also function as paracrine signals. I would like to now talk about GI hormones and the history of these hormones. These hormones in the gastrointestinal tract occupy an interesting place in the history of endocrinology. Starting in 1902, two physiologists discovered that acidic chyme entering the small intestine from the stomach causes the release of pancreatic juices even when the nerves of the pancreas were cut. Because only communication remaining in the intestine and the pancreas was the blood supply that ran between them, these two physiologists postulated and the existence of a blood-borne factor released by the intestine rather than the brain. When the duodenal extracts applied directly to the pancreas, they knew that they were dealing with a chemical produced by the duodenum. They named the substance secretin, and these physiologists proposed a general name for the hormone. And in 1905, another physiologist found the existence of the gastric hormone that stimulates the gastric acid secretion. It took more than 30 years for researchers to isolate a relatively pure extract of the gastric hormone, and it was finally named gastrin. These hormones are secreted into the blood and transported through the body. They act as the GI tract, accessory organs, and distant targets such as the brain. These hormones are secreted by isolated endocrine cells that scatter among the cells of the mucosal epithelium. There's three parts of the GI hormone families. First, there is the gastrin family. This includes hormones like gastrin and CCK. They have peptide and amino acids, ECL cells, and parietal cells, and they stimulate gastric acid secretion and mucosal growth. Next is the secretin family. This includes secretin, vasoactive intestinal peptides, and GIP. These inhibit gastric acid secretion and stimulate insulin release in response to glucose in the intestinal lumen. The third family of peptides contain those that do not fit in the other two categories, such as motilin and the GLP-1 parts of the intestine. The primary members of this group is the hormone motilin, as I said, and this increases secretion or associated with migrating motor complex and inhibits by eating a meal. I think all of these hormones are extremely fascinating, and if you have any more questions, I would read the chart for more help. Thank you so much for listening, and I hope that you learn more about short and long reflexes of the enteric nervous system. Thank you so much, and I hope you have a wonderful day.